Good morning, everyone. Hopefully everyone is doing well. We are going to finish off our lecture on viruses today. First thing, though, I want to talk about the midterm. I'm sure everybody has lots of questions about the midterm, and I know I did not quite make it through this PowerPoint, so I want to take a little bit more time talking about the midterm. So here's about where we left off last day. I had mentioned these slides are up on Moodle, and I think a couple of people actually emailed me last night or yesterday afternoon. There were some issues with Moodle. It actually wasn't Moodle. We got an email this morning from Microsoft. I guess it was some glitch they had, and so it was affecting all sorts of servers worldwide, but that should be all good now. Everything good is at the Kano end anyway. So let's talk about this midterm a little bit. It's on Thursday, and it's going to be on Moodle, and we're going to be using this lockdown browser thing. So I had sent everybody a sample quiz so they could get the lockdown browser installed and set up, and according to my records, not everyone has done that yet. So if you have not done that, you better get that done. You need to make sure that your computer and your technical, all that stuff is set up, because that is your responsibility as a student. If you don't have it set up for my class, you're going to have it set up for anatomy and physiology and many of your other nursing classes. My understanding is that everybody is going to be using it this year. So this is the first round of midterms. Then unfortunately, we get to be the guinea pigs. Somebody has a comment, they do not have the access to the computer they originally set it up on. Can the quiz be done twice? Yes, I think I have the quiz program, so you can do it like 10 times. Just I didn't know what number to set, but in case you need to redo it. So one quick tip for using the lockdown browser. Log in to Moodle using your regular browser, so Chrome or Edge or Firefox. Then click on the quiz, and then it will activate the lockdown browser. So it's important that you do that, because the lockdown browser won't let you log into iLearn. It'll just freeze. Okay, so I think a couple people were having that particular issue. Make sure you're in a place that has a good internet connection as well, obviously. So a couple other tips about this. So obviously we're not having a regularly proctored exam in an exam room, which was ideal, I think, for all of us. Unfortunately, we're doing everything online, and with the number of COVID cases floating around lately, maybe this is the best decision. I don't know. That's above my pay grade. Treat this as an in-person exam. All right, so you're going to study. When the exam comes, you're going to put all your notes and everything away. Lockdown browser is going to record you. It's going to flag any suspicious activity. I understand that, you know, some people at home, maybe they have a child or a cat, or, you know, maybe your mom knocks on your door during the exam. Those things are going to happen. Like I said, you know, any suspicious activity will be flagged, and I'll review it. And, you know, probably 99% of those cases are just people scratching their face, or their cat showing up, or something like that. So what does that mean? It means I don't want to see any papers or anything that's non-essential for the exam. You're going to have your computer ready to go, and you're going to write your exam. The only difference is it's going to be through the online system. So as I mentioned, the online system is going to have a variety of different types of questions. So you can go through the questions. I think somebody had asked me before about being able to go back and review questions. Yes, I will have it programmed so that you can go back and review questions. So you can, if you skip one, that's okay. You can go back and review them. I will have it in different sections. So I'll have, you know, the multiple choice section, and then the matching section, and so on. So I see a couple of people are asking some questions. A blank sheet of paper is okay to scribble on. Yes, a blank piece of paper is okay to scribble on. Will your mics be on? Yes, the lockdown browser will expect your mic to be on. It's not really a big deal. Like I said, if something is flagged, I'll review it, and try not to stress out too much. This is a learning curve for all of us, or we're trying to figure this out. So, if you do have some concerns, just let me know. Another question here about showing student IDs. I don't have that programmed in. I'm just one step at a time. It will definitely be something that will be required for the final exam. My understanding is that not all students have received their Kano IDs yet. So if you don't know how to do that, you'll have to go, I think it's on the Kano website somewhere. Maybe just Google it, Kano selfie ID. You're supposed to take an ID, a photo of yourself, and it gets sent in. And they will make the ID card, and then you pick it up at the registrar's office. But I'm not going to require it for this midterm because I understand that probably a lot of people have not quite yet done that. But you will definitely be required to do that for the final exam. So one more little item to add to your list of things to do. So like I said, treat this as an in-person exam. We do want to take academic integrity seriously here. I know that I do. I want to know that you are learning microbiology. I want to know that you are, I want to be confident that my exams are good, and that you are, we're all doing this honestly. So please treat this as an in-person exam. It's not the most ideal thing, but it's what the college has chosen to do. So I'm going to go with it and try to, I'm going to hope that it's going to work for the best here. Like I said, this is the first time through for many of us, so we're going to try not to be too stressed out. We're going to try to just hope it all works smoothly. I want you to focus on studying. I want you to focus on learning the microbiology. It says, so login, okay, I mentioned this already. Make sure you do log into Moodle through your usual browser first to access the test, and that will activate the lockdown browser. So make sure you do that sample quiz. If you have not done, if you're one of the few people that have not done so, get that done right away, okay? I want to know of any issues before Thursday, please. Yeah, so just to try to take some stress off, if something does not work, let's say you have a massive internet outage or something like that, we will work through this. There's been discussions in my department. If something like that does happen, what we can do is basically this midterm won't count, and we'll throw that mark into your second midterm and your final exam or something like that. I think this is what we're talking about departmentally as the protocol to do in this case. I'm not sure if that's been finalized yet, but if something doesn't work, you need to let me know right away, and we'll work through it together. Okay, so let's talk about the material in the midterm. So I told you before, this is the format that I usually use for my midterms, and it's basically the plan for this one. I might tweak it a little bit. I think fill in the blank is a little bit harder to do online, so maybe I will change that into something else. I haven't quite decided yet, but roughly this kind of format where you're doing a certain percentage of it will be multiple choice, true to false matching kind of things, and you'll that part will be graded immediately, and then there'll be a part that I'll have to grade that'll be some written. I do have a couple of questions here about the lockdown browser. If you have any particular issues with it, please email me. It's a lot easier for me to look at that one at a time. Someone says, will I be able to log into the lockdown browser prior to 10:30? I'm still working on that. I think what I will do is try to have it available a few minutes before 10:30, so people can go in and they can do the microphone and video check and all that. I'm still working with the system to see how that works. I will tell you this, that 1 hour for most of you should be plenty of time. I know there are some people that are a little bit slower and are going to need the full hour, but I expect probably 80% of the class, the exam will take about 45 minutes. So there should be a decent amount of extra time if you're a little bit slow with respondus and all that. If you are somebody that does take the 1 hour, I'm sorry. There's always people that are a little bit slower than usual. You just have to work on those skills because this is not the only class you're going to have that issue. And like I said, there should be plenty of time to finish the exam in 1 hour. Okay, so I was talking about studying last day, and just saying, you know, there's lots of things you can do with studying, and you got to think about what is going to work. And you know, I tell my students, you know, you want to work on getting memories. You want to get things stuck in your brain. So there's different ways you can do that, and there's so many different strategies. Some people love to do flow charts. Some people like to do the recipe cards. Some people do mind maps. There's apps for mind maps. Some people like to do it with a pencil crayon on a poster. There's all sorts of situations like that. But what really works is doing something creative, not necessarily creative, but something constructive where you're writing things down or you're taking notes and you're making your own notes, because that's really important to form those memories. If you're just staring at something, if you're just, you know, going on the internet and watching YouTube, that's great. There's some good stuff on YouTube, but just watching something is a lot more passive and you're not going to form those long-term memories. You need to be active. So making your own notes is really, really important. You can see my notes, the suggestions here about reviewing the study questions, the vocabulary. So that's really important that you do understand the vocabulary. That's why I have vocabulary lists at the end of every set of notes so that you can review those questions. And the study questions are really some of the big concepts that you really should be focusing on in your studying. I know there's a lot of material in there and there's lots of little nitty-gritty details, but focus on those study questions, the vocabulary, and you're gonna do okay if you have a solid understanding of those bigger picture things there. Talk to your friends, find out what they're doing. Some people do some weird things. I remember going to school and there was somebody in my hallway and she had the recipe cards and they're all taped along her wall. And I remember walking by the door and she was standing on a chair basically staring at these recipe cards, and recipe cards didn't work for me. But for her, it did, and she had them all organized, and all that time she took organizing helped her learn the material. So I see there's a few other questions here. Let me just quickly flip through them. Okay, somebody answered that one. Do we have class after the exam? Nope, there will be no class after the exam. Somebody says, can we go to the school library to take the exam? The room I rent is quite noisy. Unfortunately, no. I think we are working on that at the college. I'm not sure if that is set up yet for people that may have accommodations or technical issues. If I were you, I would maybe check with the library and see if they have a quiet room that can be booked, but I'm not entirely sure without discussing with them directly. I know the Wi-Fi has been upgraded at the college, so that's good. And there probably is some space that you can use. And quite honestly, most of the classrooms are not being used. And if you came to the college and found an empty classroom, that may just actually work. I don't know if you'd want to check with security to see if there's a room booked, but probably it'd be very easy to get lucky and find a room. Like I'm in this room here, S207, and I don't have it booked. I'm not using the normal classroom because it's further away from my office, but I like this one. So somebody's asking, it's really important to know what diseases belong to the different phylums. It's more important to know the diseases and the organisms, not the phylums. And so we'll get into that in a few minutes here. So check with the library, and if that doesn't work, like I said, you might be able to look into using a classroom for the midterm. Like I said, you know, I come to S207 here, so I know it's free during Tuesdays and Thursdays. Okay, so we covered 6 topics. We're going to just finish viruses today. So this is what you need to study. We're going to just talk a little bit about topic 7 today, but that will be not on the midterm. So let's just go back and kind of review what we talked about. Topic 1 was an introduction to microbiology. So a couple of things from topic 1 were on the quiz. So mostly topic 1, there's some definitions in there. You may remember we talked about things like commensalism and mutualism and parasitism. So there's some definitions in there, and there's a little bit of history in there, and kind of some general knowledge about, you know, good and bad things that microbes are doing to us. So a little bit in there, nothing that would be found necessarily in any of the long answer or case study type questions, but definitely a few multiple choice or true and false kind of things. Topic 2, same thing. It was a lot of this stuff was on the quiz, and much of this stuff we covered in more detail in the subsequent topics. So, for example, we talked about bacteria here. We introduced them, and then we talked about them later in more detail. So, take a quick look at this topic, but then focus on definitely topics 3, 4, 5, and 6. So here's the bacteria. We're going to just review these structures here. You can see I have them all numbered, and let's just take a look at what we have. So you can see number 1 there is that big blob in the middle, and it's the DNA, and there's some different names for it that we talked about in this class. So chromosomes 1. Remember, another term we used was, oh, there we go, nucleoid. Oops, nucleoid. Can't spell. Oh, number 2 already popped up. Oops, on that. So pilus, fimbriae. Remember, those are made out of proteins. And there is also a specialized pilus called the sex pilus, or the conjugation pilus, which is involved in conjugation. So number 3 are these little speck-like things in the cytoplasm, and those are ribosomes. So ribosomes are the site of translation, which is protein synthesis. So number 4 is one of our F words. This is the flagellum. So flagellum is 1, flagella is many. So 5, 6, and 7 are a little bit harder to figure out from the cartoon because it's, you know, we've got pink and we've got a different shade of pink and we've got a third shade of pink. But you got to look at the order that things are in, right? So the thing that is closest to the cytoplasm is definitely going to be the membrane, whereas what's next to the membrane is the cell wall, and then what's on the outside of the cell wall, that is going to be some sort of secreted material. So here are the answers. So remember, capsules or slime layers have another name. This is the glycocalyx. So that generally means sugar coat. Capsules are a little more organized. Slime layers are a little more loose and goopy. They're all usually relatively sticky because carbohydrates absorb a lot of water. There's a cell wall. Remember, the cell wall is made out of peptidoglycan, or PG. We'll just use an abbreviation there. Number 8, that should be an easy one. That's the cytoplasm. And then the last one, number 9, is a plasmid. So plasmid is kind of like a mini chromosome. You can see it's circular, just like the main chromosome. So make sure to review your bacterial structures and then some of the example bacteria that are listed in that unit. So topic 4 are the protists and the fungi. So like I have said before, don't worry about, oops, slides always are very sensitive. Don't worry about that, okay? That's the phylums. What I want you to know are these things here. Some of them have common names, right? So for example, Giardia, common name for it is beaver fever. Trichomonas, the common name is trick. So hopefully that one's easy to remember. The trypanosomes, remember there were 2. We had sleeping sickness. I'm not sure what this pen is doing, but sometimes it does weird things. And we also had Chagas disease. So know those things. This stuff here on the left, you can see I have Giardia has no mitochondria and 2 nuclei. These are just little things, you know, to distinguish them from one another, right? It's kind of like when you meet somebody for, you know, and you're remembering, okay, this guy's name is Bob, and Bob wears glasses and he has red hair. You know, those are things that help you to remember about him. So Giardia has 2 nuclei. That's kind of cool. hopefully that helps you remember a little bit about it. And of course, obviously, the disease. Somebody says loss of mitochondria, what does that mean? I mean, it doesn't have mitochondria. Giardia takes all its ATP from its host. It's a parasite. Plasmodium, remember, is the organism that causes malaria. And then we've got entamoeba. Assuming I spelt that right, dysentery, I think I put an extra E in there. And that is basically a bloody diarrhea. So sorry I didn't leave a lot of room there for fungi, but we had a couple of important ones. We had Candida, albicans. So I'm running out of room here. So that is what causes yeast infections, so thrush and vaginal yeast infections. And we also had the dimorphic fungi. I've just run out of room. But the one I'll draw it on the right here, or on the left. The one that we talked about was oops, that should be a T. Blastomyces. So blastomyces is found in the soil and sometimes rotten logs, and it's that dimorphic fungi that can sometimes get in the lungs. Okay, moving on. Topic 5, we talked about lots of worms. Remember, there were a few different types of worms. We talked about the flatworms. That was kind of the big parts of the flatworms, included the tapeworms and the flukes. So make sure you do know the life cycles for the tapeworms in particular. We did talk about that a lot in a lot of detail. The other organisms, know kind of the key things about the life cycles. The flukes, for example, remember that the flukes usually involve 2 or 3 species, and one of those species is almost always a snail. And so that's kind of important. And then the other species, sometimes you're looking at a human, or I mentioned wildlife such as deer or moose. Sometimes there's another species in there, like if you remember with some of the flukes, there's a snail part of the life cycle, and there's also a fish or a lobster. And so, like I said, kind of know generally a little bit about the flukes. They're not usually transmitted in Canada, so they're a little less important, but the worldwide burden of disease is pretty big. The nematodes. So we talked about a few there. We had the pinworms. So pinworms are, what are they? Enterobius is the scientific name. We talked about Ascaris. Ascaris lumbricoides, that's the giant intestinal roundworm. And we talked about hookworms. Very, very briefly, we talked about segmented roundworms. Mostly, I just mentioned leeches. So I'll tell you right now, I'm not going to ask you about leeches on the midterm. The arthropods, we had insects and arachnids, so we had a whole list of things. We had the insects. The big one, of course, is lice as a parasite, and mosquitoes as vectors. Of course, I'm just thinking of the other vectors. We also had the tsetse fly as well, and the kissing bugs for sleeping sickness and Chagas disease. Arachnids, so that includes things like mites, so dust mites, and scabies. We also have ticks, which are also vectors. So we're going to talk a little bit more about ticks a little bit later on in the course in Lyme disease. We did talk about Lyme disease a little bit. We are going to talk about a bit more detail later on in the course. Okay, and viruses. So it says no life cycles. I don't mean know the life cycles of every virus, but remember those 3 life cycles. We had the lytic cycle, we have the lysogenic cycle, we have the retrovirus cycle. So the examples to know, you definitely want to know the examples, those 6 examples that we're going through. I think we covered 2 of them, the papillomavirus And the herpes viruses, I think maybe we didn't quite, we kind of ran out of time there, so I'm going to just talk about them briefly again today, just to kind of recap what we talked about regarding the herpes viruses. So I do have some sample questions here. I can see my answer got highlighted very quickly. So I apologize, the initial version I put of this question was slightly different, and what I've been trying to do is, we get these test banks sometimes, and I take the questions, and I usually write most of my own questions, but I'll modify the test bank questions. And a lot of test bank questions will have, you know, E will say both A and B are true, but not C, or all of the above, or A and C are true, but not D. And I just hate those kind of things. So I've been trying to eliminate them, and I think the initial question said both A and B are true, and then so I changed E to add something else, and then forgot to change A and B. So I think this one is all good now. You can see there's a lot of different things here about the DNA. So you can see it says the DNA within a bacterial cell, and then we have a whole bunch of options. So you can see this one here is wrong because it says linear. This one here is correct. We have small circles in the cytoplasm termed plasmids, so that is good. Surrounded by a nuclear envelope? Nope, these are prokaryotes. Major structural component ribosomes? No, ribosomes are made of RNA, not DNA. And then this one also has linear, so that is incorrect as well. So make sure you read them carefully. This is a good question to easily get tripped up. And so, you know, I mean, this is the nice thing about when you're writing an exam on paper, you can cross everything out, and you're not going to be able to do this on the computer, unfortunately. So do take a look at those and read them very carefully. Like I said, if you need to, you go back and review a question. I think in the Moodle test system, there is a way to flag questions. So you can flag questions that you want to go back and review later. Someone says, do we need to know the difference between all the different DNA viruses, like the double-stranded DNA and all those examples? You need to know the examples, the 6 examples of the viruses we're going through right now. So the papillomavirus, the herpes virus, and so on. So there's 4 more that we're going to talk about here today.
Blaine?
Yes?
You said we were allowed to have a blank piece of paper, correct?
Yes.
Okay, perfect, because I'm definitely one of those people that like to write out like A, B, C, D, and then cross them out as I go. It kind of just...
I will allow a blank piece of paper for sure. I would just make sure it looks like a blank piece of paper.
Can you just hold it up in the beginning and then at the end show like what you...
That would be great. I think, like, you know, some things will look more suspicious than others. Like if somebody has a full notebook, that may, you know, look a little more suspicious, or if it's all creased and folded, it may look like it was something prepared. But if it's a nice, clean, you know, sheet of paper, that's going to certainly look a lot better.
Perfect. Thank you.
So here's a harder one. There are going to be a few harder questions like this. You can see this one here, papillomavirus. Which one is this? Like I said, I always go through my tests, and I'll put 2 or 3 marks, maybe up to 5 marks are going to be there to reward the people that have studied really, really hard. So the answer here, this is a DNA virus, and it's double-stranded DNA. There's going to be a matching question. As I mentioned, my matching questions usually have a theme around them. I'm sorry, this is not the best example. So the matching question, I think I mentioned last day, you know, it could be bacterial structures, or it could be all something to do with, you know, a theme around viruses, or transmission mechanisms of protozoan diseases, or something like that. So usually there's a theme around my matching questions, and they're going to be 5 marks. So here's an example. You can see, you know, you got to read. Usually there's a word on one side and a definition or an explanation on the other side, or if it's a you know, transmission mechanisms, it might be the organism on one side and then the transmission mechanism on the other side. So it might be transmitted by mosquito, or transmitted by being bitten by a carnivorous animal, or something like that. So we'll go through these answers here. You can see that obviously HIV is the only virus there, so that's an easy one. Again, sorry, this isn't the best matching question, but hopefully you get the idea how matching works. B, tumor, the result of uncontrolled cell division. C, a eukaryote, which is a protozoan, and then D, a prion, which we're going to talk about here momentarily. Just when we get to the end of the viruses, we'll talk about what a prion is. True or false? This one sometimes trips people up. This one is actually false. So Van Leeuwenhoek was the first person to observe bacteria. Robert Hooke was the first person to observe pork cells. So just a reminder, read carefully, and we will review the exam afterwards. Fill in the blank. All viruses contain a genome made of blank or blank. So hopefully, the answer there, it's DNA or RNA, and a protein called a capsid. So like I said, I'm still haven't decided whether I'm doing the fill in the blank. I haven't had a chance to finish kind of programming Moodle to see how easy this is to do. But I might do kind of a modification of fill in the blank, where rather than having actual blanks, I might ask you, provide 3 examples of, and then, you're going to give me some examples, and then that way it's a little easier to mark. So short answer by short answer. So sorry, this is an older question. I don't usually do these longer type questions anymore. What I do now are the case studies. And so the case studies are kind of like what we've seen in class, where it gives you a bit of information about a patient. And then there will be, they're worth 5 marks, so there'll be 5 questions. So let's say the patient is someone who, has let's say papillomavirus infection, right? So it might give us some information about that, and then ask you a few things about papillomaviruses, right? So it might ask you, papillomaviruses are naked viruses. What does that mean? And so that will be 1 mark. You explain what naked virus is. Question 2 might ask, what are some preventative measures for papillomaviruses? And so you might talk about safe sexual practices to prevent STDs and also the vaccine. And so, like I said, I'd find 5 mini questions to ask you around the case studies. So someone's asking, is the test 100% marked by Moodle? No, the written part I will be answering, and Moodle will automatically mark the matching and multiple choice parts. Okay, so yeah, found this cute little diagram on the internet, thought it was kind of cute. So, midterms don't have to be bad, right? You get to learn stuff, that's great. You get to learn microbiology, that's even better. I know it's a little stressful because you still have all your normal classes and you have assignments and things like that, but we'll get through this, and I'm hoping you all will do really well. So please let me know if you have any further questions. You can, like I said, email me or phone me during my office hours. My phone number is on the course outline, and or we can set up a Zoom meeting if you prefer that as well. Okay, so I'm going to switch now back to talking about viruses. And okay, where is it now? I just got to find that PowerPoint. Okay, so I don't think that shared, did it? Oh, there we go. Okay. So like I said, I just wanted to kind of go through the herpes viruses infections again. We kind of cut off short when we got to chickenpox and shingles and mono. So let's talk about them. Like I said, these herpes virus infections in humans, I expect you to know these 4, number 1 through 4. Don't worry about 5 through 8 and the B virus stuff, but I do expect you to know the first 4. So herpes simplex 1 and herpes simplex 2, which are right here, that cause cold sores and genital herpes. So this is an example of a latent infection. And so I think we started talking about chickenpox and shingles, and I'd mentioned that shingles is a reoccurrence of the infection. So chickenpox, it will hide in your nerve ganglia. The virus does anyway. And sometimes when you get older, or if you get ill from something else, your immune system is a little bit suppressed, shingles can reemerge. So nowadays they're looking at giving people the shingles vaccine, which is the same as the chickenpox vaccine. I think it's just a higher dosage. Last time I checked, there were 2 formulations. I don't know what the difference is between the vaccines or what is used in Alberta. I haven't looked in a while. But somebody had a comment here. Okay, I guess maybe not. I don't see anything there. So yeah, I think we talked about chickenpox. I think maybe this is the slide that I finished off on, and I was mentioning that chickenpox is spread through a respiratory pathway, and so it's relatively infectious because it is technically airborne, and so this is why if somebody has chickenpox, you keep them away from school because they can infect other people. It's not as infectious as measles. We'll talk about measles kind of near the end of the course. Measles is super infectious, and it's a lot more dangerous than chickenpox. Most people who get chickenpox, especially if they get it as a child, it's relatively minor, fever and blisters. There are sometimes complications where people get pneumonia or they end up with secondary infections from it that can be serious. But for the most part, it's not as bad as something like measles, but more on measles later. So you can see on the right-hand side, it's talking about the infection of the nerve ganglia. And like I said, this is where it can hide and go latent and hide for decades. There's somebody with a shingles rash, so that one's not too bad. That's one of the first things they look for if they think maybe you have shingles. They're looking for that rash, and they're going to wear gloves, and because it is infectious. Okay, so this is where I was hoping to start today. I guess I've been a little bit slower than anticipated, but that's okay. We'll get through this virus stuff 100% for sure, and then you'll be ready and set for the midterm. So herpes virus number 4 is the Epstein-Barr virus. So those were named after a couple of scientists who investigated this. And this virus causes, in most people, something called infectious mononucleosis, or sometimes we call it mono. You may have heard of it. Sometimes people even call it the kissing disease. So that gives you an idea how it might be spread. It's typically spread by saliva. So this virus actually infects the lymphatic tissue. And so when you get it, you can have a whole range of symptoms, kind of depending on where your immune system is at. So you can see over here on the left, it says it may cause a type of lymphoma called Burkitt's lymphoma. So I wanted to show you, why is this not advancing to the next slide? Oh, there we go. So depending on where your immune system is, you could get a full range of symptoms. If your immune system is vigorous, so for example, if you're relatively young, and so people kind of get mono very typically in kind of the 15 to 25-year-old range. Not necessarily that you're kissing anybody, but just in terms of exposure and things like that, you might get infectious mononucleosis. So it's infecting the lymphatic tissue, and people end up with fever and extreme fatigue, and you may have seen some of the other symptoms there. A really common symptom is swollen lymph nodes. So for example, I know someone who had mono, and kind of under here, under the neck, their lymph nodes were swollen so much that you could actually see the lumps. And also under the armpits. So usually a physician, when they're diagnosing it, that's what one of the things that they're looking for. I see there's a question, someone's saying that they've been anemic ever since they've had it. I have not heard of that. Usually anemic has something to do with red blood cells, not white blood cells, but there could be some relation to what's going on in the lymphatic tissue. There's a lot of things not known about Epstein-Barr virus. For example, like chickenpox, generally you get it once, and then that's it. You have immunity against it. So one of the questions that we can't seem to figure out is why we can't make a vaccine against it. It should be simple. You should be able to make a vaccine. Again, there might be some sort of viral mechanism behind it that I'm not aware of, but that's one of those interesting questions. Somebody's asking what latent means, kind of hiding and just being dormant for a while. So you can see some people are actually asymptomatic. And about 95% of us have actually been exposed to this virus and have had infection by the time you're 30 years old. And many people are asymptomatic, so they have the infection and it's very mild or no symptoms at all. You can see if your immune system is poor, this can lead to various types of lymphomas and a couple other kind of cancers. And so this is relatively common, not in Canada, but in developing nations where people may be malnourished and their immune systems are not in good shape. In fact, this was one of the earlier viruses that was connected with cancers. And I think Epson was actually in, which country was it in? Congo or Ivory Coast, where he was investigating it in a place where people were being malnourished. Okay, so I do have a case study here, and you can see the case study says we have a 20-year-old female in the emergency room. She's got a 4-day history of fever, chills, myalgia. Myalgia means some aches and pains and fatigue. 2 days prior, she had noted painful genital lesions. She had also developed a headache and a stiff neck. She admitted to being sexually active, but had always been in good health with no history of STDs. Okay, so let's take a look at these questions here. What is the most likely cause of her symptoms? So I know you're looking at this and you're going back through the viruses that we talked about. Let me just see if I have the, I guess I don't have the answers on the slide there. So somebody is saying herpes simplex 1 or herpes simplex 2. Yeah, I think the sexual transmission, that's part of one of the clues there. Papillomavirus as well, but usually not as painful. The headache and stiff neck sometimes is relatively common as well. So what type of infection is this? So if we're looking at herpes simplex 1 or herpes simplex 2, remember one of the big words we want to use is latent for that. When the blisters happen, that's the lytic part of the stage. So both of those words can apply to this. Oncogenic, no, not causing cancer. We don't usually use the word chronic because it's not necessarily happening all the time. So latent is a better description of herpes simplex 1 and herpes simplex 2. So this says there are 2 serotypes of the agent causing this disease, and we just mentioned them. Herpes simplex virus 1 and herpes simplex virus 2. So this one is usually the cause of cold sores. And this one is usually the cause of genital herpes. Although both can cause infections in the other places. And there are cases, I think I'd mentioned before, where people can get herpes simplex infections of other tissues, such as the eye, ocular herpes. Okay, so moving on. So here we go, finally where I wanted to be today. Only 45 minutes behind schedule. Well, that's okay. We're doing fine for the semester. Like I said, we will have no problem getting through the virus material today. So virus example number 3 are the Rhabdoviridae, and the example here is the rabies virus. So this is a single-stranded RNA virus, and you can see it actually kind of looks like a little bullet, kind of an interesting structure to it. So these here are zoonotic viruses. So what does that mean? So you probably know that zoo has something to do with animals. And so that's what a zoonotic infection means. It means it's coming from an animal. So another term to know is zoonoses, and that means an infection. It kind of means the same thing. Zoonotic means from animals. Zoonosis means a condition that's been transmitted from an animal. So kind of the same word. There's the life cycle there on the left, and you can see this is going in by endocytosis. So this here, endocytosis is where the membrane from the host cell is thinking it's food, and it wraps its membrane around it and brings it in that way. You can see this virus here is being synthesized completely in the cytoplasm. It's an RNA virus, and in this case here, just looking at this note here, you can see it has its own viral RNA polymerase. So it doesn't need to go to the nucleus. It's got its own polymerase. All the other stuff is found in the cytoplasm, the ribosomes, and it's perfectly fine replicating there. So what's going on with rabies virus? You probably know that you get a shot for your dog, and it's actually the law in Canada, which is a good thing. And so our dogs are not usually a risk factor for getting rabies in Canada, and we'll talk about that in a minute. But you probably know as well that rabid animals tend to be violent. They foam at the mouth, and they might bite each other. So it's infecting the brain. And you can see it's kind of mentioned there. It says the rabies virus binds and inhibits nicotinic receptors in the brain, interfering with communication and inducing frenzied behavior. I'm not really too familiar with rabies in humans, but humans can get rabies. And so the risk factor in Canada for rabies is wildlife. So you can get rabies from rabid raccoons or bats or carnivores are typically the source of rabies. Thankfully in Canada, it's not as common as other places. There are many places in the world though, again, developing nations where rabies is really common because there are feral dogs. So these are dogs that were pets, and they just wander around and they get rabies, and they wander into villages, and they might bite a child or an adult for that matter. And it's a very, very serious thing. And so what do you do about rabies? So let me just see, I thought I had another slide about rabies here. Yeah, so here are some things to know about rabies. So take a look at this. It says about 60,000 people a year die from rabies around the world. And they're usually bitten by these rabid dogs. This is hugely preventable. Even if we can't vaccinate all those rabid dogs, people can get the vaccine. The good news about rabies is that if you get bitten, and if you get bitten in your toe, it takes about 2 weeks for that virus to travel all the way from your toe to your brain. So if you do get bitten by an animal, the closer to your brain, the sooner you want to get the vaccine, but you can get the vaccine, mount immune response before it gets to your brain. So that's the good news about rabies. Like I said, the bad news is it does infect the brain, and the other bad news is that it is, in most cases, 100% not treatable. So that's why you have to get the vaccine. There's no other way to treat a person. It's 100% fatal, other than if you get vaccinated. You can see in Canada, skunks and raccoons and bats are the usual suspects, partly due to exposure. Skunks and raccoons, for example, they tend to be relatively urban, and they wander into towns and provincial parks and places like that where people might have contact with them. Not that other animals don't get them, but those are kind of the common suspects anyway. So I think I mentioned about being completely preventable. With the vaccine, it's obviously not part of our routine vaccines because most of us are not getting exposed to it. But, this is something that does happen. And if you do get exposed to it, go get the shot right away, and you'll probably be just fine. Okay, so virus example number 4 is the influenza virus. So this virus is hugely important. We're talking about, we don't even know how many infections countrywide every year. We have an idea, but most of them are not reported. In Canada, we're looking at anywhere from 3,000 to 5,000 deaths. And, if you've ever had the flu, you probably know sometimes it's kind of mild. Sometimes it's pretty serious. I remember having the flu, I don't know what year that was, a few years ago now. And, the aches and chills, and it was about 3 days of being sick, and which isn't really in the context of things too bad, but it can lead to secondary infections such as pneumonia, and so like I said, huge burden of disease. So notice this thing about these. So the phylum or the family is called the Orthomyxoviridae. A lot of these are specific to certain animals. The big one that causes the most burden of disease is influenza A, although B and C do cause people to get sick. Just there's more disease from A, and it's usually more serious. And you can see the note is it can infect some other mammals, such as pigs and birds. So that kind of makes it a little bit more of a dangerous disease because it can be zoonotic as well. So here's some pictures of some influenza viruses. You probably know that they often have these codes. So you can see there's H1N1. We were talking about that a lot in 2009. So it doesn't seem like that long ago when we had our last pandemic. You may remember the H1N1 pandemic in 2009. The good news about that pandemic is we have a system for making flu vaccines, and we were able to produce a vaccine in 6 months. And also the timing of it too was good in that you know, a lot of the pandemic was over the summer when people were not necessarily getting the flu because it transmits a little better in cooler temperatures. So what is that H1N1 all about? That is the virus structure. So if you take a look at this virus structure, it's an RNA virus. Oops, these slides love to advance to me. It's an RNA virus. It has an envelope, and that envelope has these spike proteins. So 2 spike proteins are worth mentioning. We've got hemagglutinin, and we have neuraminidase. So what are these things doing? I can't remember which is which, but one of them allows the virus to enter the host. I think that's the hemagglutinin, and the neuraminidase is allowing the virus to escape when it's budding from the host cell. So these are really essential proteins. They're glycoproteins that are essential for infection and then budding at the end. And virologists have taken a look at them and they've classified them into different families. So you can see there are 16 families of hemagglutinin. I think it's 1, 2, and 3 are the families that infect humans. 5 through 9 are infect birds. Pigs kind of have a little bit from both, and so on. Neuraminidase, kind of same thing. There's 9 families. So this is all part of the classification and what viruses do, or what virologists do when they're taking a look at classifying these things. So like I said, some of it has to do with host specificity. Some of it has to do with just how different these are. So the thing is, that means there's a whole bunch of different influenza A viruses. And that's part of the reason why we need these flu shots all the time, because there's so many of these different viruses all around. There's H1N1, there's H1N4, and so on and so on. So if someone says, is neuraminidase an enzyme? Yes, A's means enzyme. And like I said, I'm pretty sure that's the one that's involved in allowing the virus to bud. So I think what it does is it clips something that allows it to escape from the host cell. So here's another picture from the same source. I think this is the CDC, and they're showing the flu infection, and you can see, yeah, so there we go. It looks like the hemagglutinin is recognizing this thing here. It's called sialic acid. Maybe you can see that up there. Sialic acid is just a carbohydrate that's found on the surface of our cells. So in humans, sialic acid is found in our respiratory tract. So influenza, you probably know, is a respiratory infection. So that's all down our throat, down our bronchi, in the lungs, and so the infection can get in there. The deeper the infection, the more dangerous it is. So I want to talk a little bit about what is special about influenza viruses and why we are getting a new flu shot all the time. Every year there's a new flu shot. And the reason for that is something here called antigenic drift. So take a look, here's my little flu virus. My little flu virus, you can see it's got some RNA there in the middle. It actually has 8 strands of RNA. 10 genes, 8 strands of RNA. I don't know how that works or why they don't just do 10 strands, but hey, I didn't put it together. I'm not Mother Nature. It has a capsid. So you can see the capsid here is these little blue proteins. And then on the outside is the envelope, and you can see there's the neuraminidase and the hemagglutinin. So what is antigenic shift? So it turns out one of these 10 genes is RNA polymerase. And the viral RNA polymerase is sloppy. So it's not very good at copying those nucleotides. And so this virus mutates. And the mutation rate is kind of enough that we get a new strain every year. So if you take a careful look at my diagram, you can see my hemagglutinins, they're kind of square over here. Over here, they're kind of round. So they just kind of change slowly. Some years our immune system will recognize the old one quite well, other years not so much. And if we're lucky, we have mild disease. If we're not so lucky, we have more dangerous disease, and a lot more serious. This is one of the reasons why when the H1N1 came out in 2009, they had realized that younger people were at risk, which is kind of a weird thing to happen with the flu virus, because younger people had not been exposed to anything that was even remotely like that H1N1. Whereas older people, I think the age was over 50, some of those people had been exposed to a very similar virus, 50 years previous. And so that's part of what's going on with our immune response. So antigenic drift is slow random mutations. So when we make our flu vaccine, usually what we're doing, it takes about 6 months to make a flu vaccine. So we're trying to have our flu vaccine ready usually for around October-ish. So that means we go back, and if you think 6 months previous, we're looking around February, we're making some decisions. And what is often done is people are looking at the flu season that's going on in the southern hemisphere. So places like New Zealand, because they'll have a flu season in their winter, which is our summer. And so it's kind of a little bit of a guessing game as to which strains to use and which ones they think are going to be most prevalent. So usually the flu vaccine has 2 to 3 different strains in there. I think there was even one year it had 4 different strains. And like I said, it's kind of a guessing game to figure out which ones are coming. And then when we have our flu season, the New Zealanders are looking at what's going around in Canada, and that's the vaccine they're going to make. So like I said, antigenic drift, slow random mutations. Okay, this is something that happens all the time. Maybe some year we'll figure out how to make a universal flu vaccine that protects against all flu strains. There's a lot of people working on that kind of thing, but at the moment we're out of luck. Some years the vaccine is really effective, and other years it's not, partly because, like I said, to do with that guessing game of which strains we think are going to be the most common strains in the human populations. So there's a second thing here I want to talk about called antigenic shift. Right, so I mentioned H1N1, and what happened there? What happened in 2009? Why, what was going on there? So antigenic shift is where we have a little something else going on. So take a look here. I have 2 different flu viruses. There's the human one at the top, and I have some sort of avian strain on the bottom. So avian means bird. So sometimes, this is very rare, 2 different flu viruses can infect the same cell. So you can imagine that's not going to happen very common. And humans don't usually get infected by avian strains. But unfortunately, pigs can. Pigs actually have the receptors for the human flu viruses and the receptors for the bird flu viruses. So this is why some people were calling it the swine flu. Not entirely accurate, because it had human virus parts and avian virus parts, so it's kind of was a bit of a blend of these things. So sometimes, very infrequently, 2 different flu viruses can affect the same cell. So that is what happened just before 2009. I think it happened in 2007, is we had these 2 strains. Actually, it was 4 different strains infect one cell, and what you end up with is a reassortment. So remember, I was just saying that we have 8 strands of RNA. So we have 2 viruses infect the same cell, and it kind of mixes and matches. Okay, we'll take a strand from here, and a strand from there, and a strand from here, and a strand from there. And now we have, well, what do we have? We have a brand new virus. And this brand new virus is unlike anything that has ever been seen before. And this is why it is potentially dangerous. Because this virus, we have no immunity. It's going to infect humans, and people, of course, are thinking about what happened in 1918 when we had a massive flu pandemic worldwide that killed millions and millions of people. And so this is what happened in 2009, is this happened, and we had a brand new strain. And of course, like I said, people were thinking about the Spanish flu of 1918. And the good news is it turned out that the H1N1 in 2009, most people did not die from it. Some people did, and younger people got sicker than older people, which is kind of weird around the flu. Like I said, when you have a new virus, this is what happened. But thankfully, it wasn't as bad as the 1918 flu, where millions of people died. So we'll talk a little bit more about pandemics later on in the course as well. So flu, as I mentioned, respiratory symptoms. You're looking at a cough, chest pains, typical virus symptoms, so fevers and aches and things like that. And as a respiratory virus, we've been talking about these like a lot lately. So what do? You wash your hands, you cover your mouth. You can see this is from Health Canada. They're recommending when you sneeze, you cover your mouth like this. So you're putting your elbow there, because if you cough into your hands and then you're touching the doorknob or whatever shopping cart at the grocery store, you're going to spread the virus. And of course, nowadays we're talking a lot about masks, and we'll talk a little bit about that later on. I can see somebody has a question about whether the antigenic shift can happen with COVID. So the SARS coronavirus 2 does not have those multiple genomes the same way that the flu virus has, or multiple strands of RNA. It is an RNA virus. It just doesn't have all those strands. So it can't do an antigenic shift. It can do an antigenic drift, but not at the rate that the influenza, because it turns out that the SARS virus has an RNA polymerase, and that RNA polymerase actually has a proofreading ability. So there are mutations out there, but at the moment, the current data seems to show that the mutations, as far as we can tell, they're not significant. You may hear in the media, there's been a couple of our media reports about it's mutating and it's more dangerous or it's spreading better. Honestly, the science isn't there yet. We don't know. And the bottom line is this virus is doing just fine. It doesn't need to mutate. It's infected millions of people worldwide. It doesn't, mutations at the moment. Who cares? This virus is doing really well and propagating itself. Time will tell, though, in terms of what the mutations are doing. Maybe this virus will better acclimate itself to humans. There's many possibilities. At the moment, it's mostly speculation. What we do know is it will not mutate like the flu, because it has that proofreading ability. Okay, speaking of coronaviruses, here we are. So like I said, COVID is the disease that we're talking about. That's a coronavirus disease. 19 is for 2019, when the disease was first recognized. SARS-CoV-2 is the name of the virus. So SARS was a disease recognized in 2003, which stands for Severe Acute Respiratory Syndrome. And so this virus, the SARS-CoV-2 virus, CoV is coronavirus, is very similar, I think 90% similar to the original SARS virus. So now we're calling them SARS-CoV-1 or SARS-CoV-ASIC, or SARS-CoV-2 is the name of the virus. So coronaviruses are RNA viruses. They are enveloped, and you can see a little picture of one there, and you can see this note about the origins of these things, which is actually really fascinating. And I got a little slide in the origins of these things in a moment. So there's the SARS virus. You can see it's recognizing a receptor called the ACE2 receptor. And so this is why it's, for the most part, a respiratory infection, because the ACE2 receptor, again, is something that's found in our respiratory tract. So here is the life cycle, and it looks like it is going in by endocytosis of some sort. Again, we have a virus that does not need to go to the nucleus because it has its own RNA polymerase. So I want to talk a little bit about the origins of these coronaviruses. Up until recently, very few people really were talking much about coronaviruses, other than people who are interested in such things. And I'll kind of, I'll touch on some of those aspects. So it turns out there are four common, they're usually called the cold coronaviruses in humans. And turns out we don't actually know a lot about them. You can see this is showing that these originated in various animals, so bats or rats, so rodents, and often they seem to go through some sort of intermediate host, meaning they mutate just a little bit and then eventually enough to infect humans. And at some point, these all jumped to become common human infections. And we all get these. We all get them when we're kids. You get your runny nose and nobody thinks anything of it. And then every year, most of us get these. viruses again and again. Every 6 months to a year, we get infected by these viruses. Often we don't have symptoms. Often it's just cold-like symptoms. Sometimes we end up with something a little more serious. But for the most part, these viruses have not really had a lot of study because they're not really killing a lot of people. They're cold viruses. Like I said, you know, the interest hasn't been too high until the last year, where suddenly people are very, very interested in coronaviruses because, of course, you know, we're in the middle of this pandemic. So there were other viruses that we've documented that have crossed over to humans, and we're still considering these ones zoonotic. So one of them is the original SARS Classic. That was in 2002, more 2003 if you look at most of the human infections. And you can see that is coming from a bat. We found very, very similar viruses in bats. And the intermediate animal is something called a civet. So if you don't know what a civet is, that starts with a C, I think it's C-V-I-T. It kind of looks like a cat slash raccoon. This caused SARS, the SARS epidemic that happened in 2003. And this was very, very scary. I know many of you were probably not that old in 2003. I remember it very clearly. This was killing a lot of people. This killed something like 40% of those who were infected. The good news was people had symptoms right away, and it was easy to isolate those people. So this actually, part of the epidemic ended up in Toronto, where I think it was 40-something people got infected. And thankfully, it was isolated very, very easily. The good news, we haven't seen it again. Never again has that jumped from animals to humans. There's been a couple of, I think, cases where somebody studying it got sick from it. But other than that, we've never seen SARS-1 again. So my question is, for all the people studying coronaviruses out there, what have you been doing for the last 20 years? Clearly, this could have happened again, and now it has happened again. Another SARS virus has jumped to humans. Thankfully, most people are not getting severely ill, but it is quite serious. You know, we're looking at, I believe, worldwide, we've crossed more than 1 million deaths now, which is huge. If you look in Canada, I think I mentioned we get about 3,000 to 5,000 deaths from the flu in Canada per year, and I think we're at something like 10,000 deaths from SARS-CoV-2. So this is relatively serious. There's another virus. You can see MERS. What does that stand for? I think it's Middle East Respiratory Virus. Has also crossed to humans, and this is actually also very, very serious, and it seems to be associated with camels. So there's been outbreaks in Saudi Arabia, I think South Korea. I'm trying to remember. There's a handful of countries in Asia where this has happened, and like I said, it's relatively serious. And actually, we've learned a little bit from this because people have actually been working on MERS vaccines, and this has been a starting point for some of the human vaccines for SARS-CoV-2. So where did SARS-CoV-2 come from? It seems that it also came from a bat. There was a virus that was isolated from a bat a few years ago that is something like 98% similar, very high percentage of similarity. It crossed over to humans, and some people are thinking it came through a pangolin. So what's a pangolin? So Google pangolin. They're very cute. They're these weird little mammals. They're found in Asia. They have scales on their skin, and they are, my understanding is the scales are used in some forms of Chinese traditional medicine. And so you have that human to animal contact. We're not 100% sure yet. Maybe we'll never know. But this seems to be where SARS-CoV-2 came from, at least from a bat originally. Could have been another intermediate animal. I know somebody else was suggesting snakes, but I think that's been, at least it doesn't look like it was the best hypothesis. So you probably have heard tons about these coronavirus diseases, and so I'm not going to get into sort of the common knowledge as much on these, but you probably know that you know, the main symptoms, of course, are fevers, shortness of breath, and coughs. There's a huge list of symptoms because it turns out that this is actually one of the most surprising things about COVID-19 is the range of symptoms that people get. Some people are ending up with blood clots. A lot of people, like something like a third of people, are losing their sense of smell and their sense of taste. So there's some weird things going on here. But again, mostly respiratory symptoms and mostly common viral type of symptoms, so fevers and chills and things like that. Some people are ending up with a second phase of symptoms, which are more immune kind of response symptoms, so fevers and something called a cytokine storm. That's something I'm going to cover a little bit more later on in the course. Okay, I realize we're running out of time here, but I do want to finish this, and we should be in good shape. So I found this chart here on the internet, and you can see kind of comparing some of the symptoms of colds and COVID-19 and influenza. So you can see there's a lot of overlapping symptoms, right? Fevers and coughs and things like that. So if you're interested, you can take a look at this. Okay, so the last virus I wanted to talk about is HIV. HIV is a retrovirus. And so we talked a little bit about the retrovirus infection. And I'm going to look at the HIV infection in a bit more detail. So you can see HIV is infecting a specific type of white blood cell. They're called lymphocytes. They're called actually T helper cells, and or sometimes they're called CD4 cells. So these are a really important part of our immune response. And so as these cells die, this can lead to a failure of the immune system. And so the disease, when the immune system has begun to fail, is called AIDS. So not everyone with HIV has AIDS. You only have AIDS when your immune system has failed. So with treatment, you may be HIV positive and never actually have AIDS. So here's the virus. I want to talk about two enzymes in particular today, the reverse transcriptase and the integrase. And you can see it's an envelope virus. I think I showed you the structure before. We have these glycoproteins. So GP is glycoprotein, and those are involved in the infection process. It's got RNA in there. You can see the reverse transcriptase is actually found packaged up with the virus particle. So I have this video here. I'm going to skip this video for today, just in the interest of time, but I will. You can access it through the PowerPoint very easily. So take a look at that. But what I want to do is just talk about the life cycle here for a few minutes and kind of point out a few things. So the life cycle starts in the top left. You can see we have an attachment event. So right here where we have those glycoproteins recognizing those CD4 receptors. It goes in by fusing, so the membrane of the cell fuses with the envelope of the virus, and then it goes into the cytoplasm and it gets uncoated. So you can see right here we have our viral RNA, and those little red dots are the reverse transcriptase. So over here, we're going from single-stranded RNA, and we're ending up with double-stranded DNA. So that's what the reverse transcriptase does. It makes the single-stranded RNA into double-stranded DNA. Like I said, that's kind of backwards from transcription. Normal transcription goes from DNA to RNA. In this case, we're going from RNA to double-stranded DNA. So it goes through here into the nucleus through something called the nuclear pore, and then it integrates into the host genome. So the integration is done by that enzyme called the integrase. So it goes into the host genome, sits there for a little while. This is something we don't really understand. Some people it goes dormant longer, some people it's shorter. Why it would, whether there's some sort of activation signal, I don't think we fully understand that. But you can see eventually the host DNA is transcribed and it makes RNA. RNA goes out of the nucleus, and RNA is used by ribosomes, and we're going to make new virus particles, and eventually the virus buds off and, in some cases goes and infects another cell. So I know I have a couple other pictures here to show you. We will be talking about HIV kind of on and off over the semester. I want to talk about a little bit about transmission of it when we talk about epidemiology. I'm showing you this map here because, I'm going to show you another map here in a moment. That prevalence of AIDS and HIV in different parts of the world, there's a huge differences, right? So Canada, you can see, is green. So that's much less than 1% of our population is HIV positive, which is great. That's good for us. Other parts of the world, it's much, much higher. 5–10% in some areas of the world. And we're going to talk about, why that is later on in the semester. So stay tuned for that. So the last thing I want to talk about is something that is not a virus, but this is a great place to fit it, is an infectious particle called a prion. So somebody is just being clever here. So P-R-I for prion, and on kind of means particle. So what is this disease? You may have heard of mad cow disease. This is what prions cause. There's different diseases. You can see there's a sheep there with a disease called scrapie. And so what is scrapie? Well, this is a neurological disease where it's affecting cognition, and in humans we're talking about things like dementia and all that. Anyway, the sheep, they get this cognitive disease, and they drag themselves along the fences. And so they're scraping off their wool. So it's called scrapie. Obviously, this is undesirable if you are a sheep farmer because you like their wool. So the human disease is called Creutzfeldt-Jakob disease. The cow disease is called bovine spongiform encephalopathy, or commonly mad cow disease in the media. So let's talk about what this is. So these are proteins that have gone bad. Hope you like that joke. I'm just looking, there's somebody has a question here. People are just laughing at my jokes. Yeah, somebody's talking about being in London in a class trip in 1996, and it was huge over there. People afraid to eat the beef. And I'll come back to that in a moment with my map. So what are these proteins going bad? So this is the protein, PRP. So PRP actually stands for the prion protein. What does it do? We don't know. It's found in our immune system, or it's not our immune system, our nervous system is found in our brain. So it's doing something in there, but we don't really know what it does. So sometimes it kind of gets misfolded. So I'll show you in this next slide what we think is happening here. This is very hard to study because this takes 20 to 50 years for disease to manifest itself in humans. So how do you study that, right? So this is from animal models and other models. We can see over here, we've got the normal protein is the square. We've got the prion protein is over here. So what we think is happening is that the prion protein causes the misfolding of the normal proteins. And during the misfolding, these things start to aggregate. So you can see we have groups of 2 over here, we have bigger groups over here. So these things aggregate and they form these things that we call plaques that are found in the brain. And you may remember the cow version of the disease, the word spongiform in there. So the brain gets kind of spongy. I'll show you some brain pictures here. So there's a normal brain there. And you can see here's Creutzfeldt-Jakob disease. Here's scrapie in sheep. And then we've got another disease here called Kiru. That's another interesting one to look up. It was associated with, believe it or not, ritualistic cannibalism in some tribes in Papua New Guinea. And they were eating the brain of people that were sick, right? And so this is partly how we think we get it. Some forms are heritable, some forms are transmissible, meaning if we eat contaminated meat, it can induce that misfolding in us. So this is the big risk, right? Is if we eat cows with mad cow disease, are we going to cause disease in humans? So usually I ask everybody at this point in the class, what happens in Alberta if a cow gets mad cow disease? And people have lots of interesting answers. And depending on the circumstances, in some cases, the farmer shoots the cow and buries it in a hole and tells nobody. And why would they do that? Because you may know when this happens, it's actually been a while since this happened in Alberta, but one cow with mad cow disease, the entire herd will get killed because you can't tell if the animal has it unless you actually dissect the brain. So they're concerned that the entire herd has it, so they will kill that entire herd. Meat does not go across the border. The Americans close the border to Alberta beef until they think the crisis has been averted, and so it's a major, major event because nobody wants to eat the meat. They're all terrified of getting mad cow disease, or in humans, Creutzfeldt-Jakob disease. So this is showing that there are some different forms of it. I'm not going to really go into that in any detail, affecting different parts of the brain. Here's the good news. The number of cases has gone down. quite a bit. And you notice that they're talking about the UK. The UK is where there were a lot more cases documented than anywhere else in the world. So this brings me to my last map and my last minute. I promise I will be done here in 1 minute. Where do we find cross-celiac disease? In the dark green areas. So interestingly, this map is kind of the different image from that map I showed you with HIV and AIDS. So what was going on way back in the 80s and 90s? So it turns out that at some point, I think it was in the 60s or 70s, some nations started this practice. So you can imagine you have a cow and the cow dies of, you know, whatever. Doesn't really matter what the cow dies of. And you're looking and you're thinking, well, that was an expensive animal and that's a lot of protein there. So the practice was you would grind up the cow and add the protein to the cow feed. So now we have cows eating cows, right? And so this is how it was getting transmitted from, you know, cow to cow and then eventually to humans. And of course, it took us a long time to figure this out with these incubations being like 20 years or more. And eventually we figured, you know, we figured out what was going on. And so most of these nations now, that's kind of a banned practice. We don't do that anymore. So like I said, this takes decades to kind of trickle out. But the good news is the number of cases that are diagnosed are you know, they're really, really small. In fact, I don't even know what the numbers are in Canada, but I think there's less than 10 cases diagnosed annually, something like that anyway. Okay, so I realize we're at the end of our time, and that's actually pretty much the end of my slides. I think there's one or two more that are, but it doesn't really matter. At this point, this is a good place to finish. I see somebody has a question here. Somebody says, do you by any chance have any Kahoots we could use on our own for studying? I can certainly share with you the ones that I have made. You can play with those ones. I had meant to make one for this virus topic, but I never got around to it, and clearly I ran out of time. So anyway, that's the answer to that question. So like I said, we're out of time here, so I will see some of you on Thursday. So again, if you have any issues with the lockdown browser, I need to know right away. If you haven't got on there, I need you to get on there right away and make sure you can get your technology to work. So good luck with the exam. Forward me lots of questions. I'm expecting lots of emails in the next couple of days for anybody who has questions. Have a wonderful Tuesday.